So first I want to thank the organizing committee for giving me the opportunity to talk in front of this group. I'm going to talk with you today about single cell writing and single cell editing. So the purpose of Muse Bio is to really inspire and enable new biology. But we realize we're not going to be the ones doing that. It's going to be our customers, really you and the audience here. Our contribution on this journey is going to be to make the process of writing single cells simple, efficient, and robust. It's really our mission to make better tools for researchers to more efficiently explore the genome. So I'm a mechanical engineer, and I have committed my life to building better tools for biologists. Before this, I started a company called Quantalife that does digital PCR, sold it to Biorad. And then I started a company called 10x Genomics, which does single cell sequencing and single molecule sequencing. I've really committed my life to building better tools for biologists. And those two previous companies were focused on better tools to read the genome. The focus now is to build better tools to write the genome. And that's really why I'm here, because I can't do this alone. I'm a tool builder, not a tool user, and I need to partner with this community. So I really mean this when I say I want to engage with you. I have tremendous support behind me from an investor class and a product development infrastructure to drive products into the marketplace. But I need to partner with the community to build the correct thing, and that's why we're here. So I specifically want to give you an update on some of the data and then an update on product development. So Muse is a kind of genome engineering tool company. The product line that we offer will be a full suite of software, instruments, and reagents, specifically to write single cells, where each cell you can put in the mutation that you want at various locations, where also we're inserting barcodes so you can track these cells. And this is a large population of cells that we'll be creating in cell variants. We started in 2015 out of the University of Colorado. 6 million Series A, and then just recently closed a 23 million Series B with the lead investors of Intel and Illumina, Venrock. There was a press release yesterday. We just announced John Stoppelnagel, who's the founder and CEO of Illumina, to be the chairman of our board. So we're very excited about committing that effort that went into reading the genome and better tools to now focusing on writing the genome. This is really the perfect audience. So really the key accomplishments that we have today, and what we have is, think of it a early stage integrated breadboard prototype system where we have the software, instrument, and reagents, talking to each other, and I'll show you some of that data. The chemistry itself was recently published in Nature Biotech called Create, and think of that as the early stage, first generation of what we're going to be branding as Forge chemistry that we'll be putting out into the marketplace. So pretty much if you think about genome engineering today, and kind of personally as an engineer, the unit operation that I see just consistently being moved around is the 96 well plate. And I've learned you need to build your infrastructure to fit with that because of the large infrastructure in this industry. But the 96 well plate, from a partition perspective, again, my passion is to trying to digitize biology. You're limited with the well just because there's not enough partitions there. If you want to do some specific edit, you put your guide RNA homology arm in, you use the well to co-localize those reagents to do your particular edit. And whether you use a library of guide RNA like the Gecko libraries, or from the Broad and Church, or a library of your homology arms like Shenduri, to try to increase the number of edits per experiment, you're still somewhat limited by that plate. So I'd like to challenge, don't think about using the well as the partition. Let's use the cell as the partition, okay? And in using the cell as the partition, you can now put, almost like a 100,000 well plate within a 96 well plate. So now you have a plate within a plate concept. Simple idea, okay, makes sense. But now how do you get the reagents co-localized so that you have your guide RNA and homology arm to do your particular edit always go into the right cell? Again, with the previous two companies, I found the best solutions that one actually can build real scalable products into the marketplace are the simple ones. So let's simply covalently attach the guide RNA and homology arms. So now you can co-localize the delivery of where you're going to do the cut and the paste. In that Forge chemistry, we also want to make sure that you can track this stuff and sequence on the back end. So we also insert barcodes within the biochemistry. So data, we're a very data-driven company. I'm going to give you 3 quick examples, building up in complexity, starting at the protein level, then moving to pathway, and then doing full genome editing. So here we took FolA, it's an essential enzyme, has to deal with antibiotic or antibiotic resistance in microbes, or in bacteria specifically here. Because we rationally design every single variant, we made 3,140 variants fully saturating this protein, FolA. And then we screened its activity, which variants were viable, and then also looked at various set of configurations here, which shown on the right-hand plot, the ability to grow in ever-increasing concentrations of this particular antibiotic. We saw a 200x increase for one of these variants, and why I think this is important is because in pulling from the literature, it's a very well-studied protein, but we found novel variants in here because we're doing full saturation of the enzyme. It literally is every single combination of the amino acid sequence that could actually go within this protein. So let's jump from one gene now, because we can control every single variant that we create rationally designed, and go to 19 genes. Here now studying a pathway. We made 16,300 variants, and in this pathway, lysine, it's $1 billion molecule. We want to increase the flux through here, and because now you can make so many variants, you can start to do some of the design on the front end different. You can somewhat coarsely grab anything that you might think that would relate to that pathway and throw it into your front end design. So we did that here, and then used an anti-metabolite screen to see which variants actually. Sorry, I was reading a sign. See which variants which variants could exist within this higher concentration of the anti-metabolite. And at the end, we saw a greater than 1,000x increase. We're doing mass spec verification that we're actually producing increasing amounts of lysine through this pathway. So let's take it up a notch and go from 19 genes to 177 genes. Here's a regulatory network within E. coli focused on antibiotic resistance. We made 75,093, don't forget the 93 variants, because again, everything's rationally designed and completely programmed right on the front end. Targeted mutagenesis at all binding pocket motifs within these genes. Did the screen and across a variety of different antibiotics, here specifically shown a set of variants with 64x increase within this pool. You can see in the bottom left-hand coverage, the cover across the chromosome that we're doing. Bottom right-hand corner, I want to remind you, is every single gene still has that first protein level resolution where you can click on and go down to the single amino acid level, and you can know which variant gave you what phenotype or what property, because again, everything's barcoded. So this quote, to me, is one of the reasons why I'm super excited about Muse, is because I really think it is such a unique moment in time that we are, as a community, able to be here to go after truly exploring an organism's genetic landscape. And if this excites you, if you have any ideas how to use this technology, this quote in the blue box gets you going, I really would like to reach out and hear from you. The way we're going to package this is and deliver it to the marketplace next year is through a full suite, again, a total solution where you have the software, the instrument, and the reagents. And what the software will do is you'll select your pathway, you'll select your protein, what you want to do, and we'll use our proprietary design rules to take that mutation that you want and translate it into what the architecture needs to be for our forge biochemistry. Then the reagents will show up. You'll have the instrument on site. You'll take, if you're going to make 10,000 variants, for example, take your 10,000 cells of a single genome, put it on the box, put the reagents in, hit go. The next day you'd come back and you'd have 10,000 custom programmed genomes specifically to the reagents that you ordered. So in closing, I just want to really drive home, I mean what I say, the email at the bottom, bd@musebio. If you have any interest in this, please reach out. Within the development cycle, there's a time window that we have to influence the configuration of the product before we release it into the marketplace. And I want to make sure I'm doing all this work so that it's useful to this community. So please reach out. We will be doing our second early access program at the beginning of next year. So if you also have any ideas, it will be limited in scope. Please send those off too. Thank you for your time.